Hello, my name is Davyd Bohdan and I am a PhD student from Moscow Institute of Physics and Technology. I present ARTEM as a tool for searching motifs in RNA 3D structures. The spatial structure of RNA is crucial for determining its functions. An example, IMANO3, a fluorescent aptamer that contains multiple non-canonical interaction red lines within its functional loop that looks like empty as a part of secondary structure. RNA's 3D structure is modular and can be considered as a composition of building blocks that we called tertiary motifs. The stems, king turns, base pairs, aminors, etc. are all examples of tertiary motifs. Some of these motifs can appear as a part of more complex motif on different ways, in different contexts, as the entire structure can be represented by composition of such recurrent 3D motifs. The distance between motifs may not be clear-cut, so the challenge we face when investigating RNA 3D motifs is how to look for them in RNA structures. For the task, let's think. Can we search for motifs by sequence? If so, an example, taking a minor as a reference, we will not find U minors, in which uracil replaced adenine, but the motif has a similar set of interactions. Does it mean that we can search for motifs by interactions? And here we have a pair of D-loop-to-loop-like motifs that form between two distant loops and has a similar topology. But the first one have long-range base pair and the second are not. So it means that if we take one of these and search for, we will not find another. But they still share the same topology. Can we search for motifs by topological similarity? And here also we have a pair of king turn and king junction motifs that, as we know, share the same sharp king at the backbone, but the first king turn is an internal loop and king junction is a multi-loop. So again, taking one of these, we will not find another. Now we see a pair of visually similar motifs. They have 3 base pair here, 3 base pair here, 3 adenines behind here, and 3 adenines behind here. But if we look on the details, it appears that this pair of motifs are not similar, because they differ in sizes. Like this strand formed by 3 residues, this strand formed by 4 residues. They also differ in base types. The sequence of base color here is a yellow, red, yellow. The sequence here is a green, green, blue, red. They differ also in interactions, because the lower base pair are not the same as the lower base pair here. The inclination angle of 3 adenines here are not the same as here. Moreover, this pair has different topology. Here we have a continuous sequence of 6 residues. In the right case, we have a 2 strand of 3 residues. So this pair belong to all 3 listed cases before, and how we can match them without knowledge correspondence between residues in advance. This problem have factorial complexity, and to solve it, we develop ARTEM for sequence topology independent and annotation independent superposition of structures. The ARTEM based on the assumption that in good superposition of 2 structurally similar tertiary motifs, at least one residue match has an RMSD close to 0. To be able match residues between match residues with different types of base, ARTEM use 5-atom residue representation. So we can perform superposition like this. And with superposition of residues, we can superpose the whole entire structure. Yeah? So ARTEM take as input to structure and perform superposition by each possible pair of residues between them. So we have a quadratic complexity. And here is the example of such pair of residues and their superposition. On the next step, ARTEM. Calculate the set of mutually closest residues in superposition in structure that is superimposed. The third step is a correction of the superposition in accordance to this set of mutually closed residues. Now we just need to select better one with some of metrics like RMSD or size of mutually closest residues set. To demonstrate capabilities of ARTEM, we take a D-loop-T-loop motif from tRNA and perform searching for structurally similar motifs in long-range LoRa dataset. LoRa is our dataset of long-range RNA modules. Long-range RNA modules formed between distant loops and helices distant in secondary structure. So the D-loop-T-loop is an example of long-range RNA module, because they form by D-loop and T-loop and have a pair of two base pair UG and CG. As a result, we find 32 D-loop-T-loop like motifs in different types of RNA, such as tRNA, yRNA, several ribose switches, and ribosomal RNAs. On the left side, we select a conserved part of reference. On the right side, we perform superposition of all finding motif on the reference. We can see a high density of each bin of residues at each position. Also, we can see by color diversity of base type on each position. Let's look on the examples. Here one has alternative residue arrangement. Instead of long-range base pair, we observe U233 that correspond to guanine and that stacked under U209, which don't allow form base pair with U206. So this uracil 209 corresponds to this adenine, and we have another residue arrangement. This case we can find with rely to independent if we rely to interaction between structures, we will not be able to find such motif. The second example of tetra loop show us that ARTEM can find similar motif in topological independent manner. Here we have instead long-range base pair a local base pair. So we find ARTEM is a powerful tool in motif searching problem. They can be useful in comprehensive analysis of RNA structures. For example, we can take a set of different motifs and perform annotation of structures. So also we try to apply ARTEM approach for global structure alignment of RNA sequences. And what we get? We develop a new tool called ARTEMIS that ARTEM based, and compare them with state-of-the-art US align tool in structural alignment RNA sequence problem. We take 500 RNA chains and perform pairwise alignment by ARTEMIS and US align. Each result we estimate with TM score of smallest RNA chain. TM score is a measure of global structural similarity. So we drop our results on this scatter plot in which each dot corresponds to a pair of TM score obtained by Artemis on x-axis and US align on with y-axis. So the dots under diagonal correspond to higher value and better alignment of by performed by Artemis. Moreover, it's known that global structure similar TM score of 0 greater than 0.45 correspond to global structural similarity pair motif. We have see many cases that ARTEM can detect. Also, we know the similar pair of structurally similar chain can contain permutation, and as we do here, because ARTEM and ARTEM is topologically independent, we allow to Artemis find and perform alignments with permutation, and this give us better results. Now we show you a couple of examples in which Artemis is success. Here is a pair of two tRNA chains. One of them missed many fragments, and even in this case, Artemis find the global structural similarity with TM score equal to 0.5. On the right side, we see this alignment prepared by US align. So in this case, US align is failed. The second example is yRNA chain and tRNA chain. It is known that yRNA chain mimic tRNA, and even in this sample, in this pair, Artemis in easy way find global structural similarity. But if we take a look at beginnings of our chain, we can see a permutation in alignments. Also, we can see it on the graph alignment with these fragments that permuted. On the right side, we see that US align also failed. So our approach that we use in ARTEM and Artemis is powerful and useful in searching motif in local and long-range motifs and one-liner, and also can be useful for finding global structural similarity. But the main approach that we use is can be applied not only for RNAs, because this approach based on the pairwise matching of molecular monomers. We can in easy way extend Artemis for amino acids also, and perform alignments and do superposition of structure and structural molecular complexes and so on. But all what we need is at least 3 atoms to represent our monomers. So for this work, thanks to my supervisor Evgeny Paulin and Janusz Bunitsky for helping. Thanks to ISMB and NCN for funding. You can watch. ARTEM and LoRa are available as a GitHub repositories, and For more information, look our article that published in bioarchive that also be accepted for publication in NAR. So thank you for your attention.